हेलो फ्रेंड, स्वागत है आपका एक और वीडियो में। We'll be continue to discuss the remaining questions of CUCET 2021 Part B के questions. So let's begin. Which of the following is not true for master regulatory transcription factor? It must be expressed when the specification of a cell type begins. It must regulate the expression of a gene specific to that cell type. It must be able to redirect a cell fate to this cell type, or must be expressed when the specification of a cell type ends. Master transcription factors, these are the topmost regulators of gene expression, and obviously they should regulate the basal rate of transcription and can begin the transcription of a particular gene. Definitely they should be expressed before a decision need to be made about a cell lineage, rather than when cell is already differentiated. Hence, a transcription factor must be expressed when the specification of a cell type begins, rather than when the cell type's specification ends. So option D is the incorrect as far as master regulatory transcription factor is concerned. So here option D has to be the correct answer. Difference between chromatin regulation by high and low CpG promoters is: high CpG content promoters repress gene by DNA methylation. Low CpG content promoters repress gene by histone methylation. High CpG content promoters repress gene by histone methylation, or low CpG content promoters repress gene by histone acetylation. Actually, based on the CpG content, mammalian promoters could be either high CpG promoter or low CpG promoters. Cytosine in the CpG nucleotide can be methylated to form 5-methyl cytosine. Such methylation event suppresses DNA activity and may change gene expression. This activity of DNA methylation is often found in high CpG promoters, which are present in regulatory genes. These regulatory genes of having high CpG promoters, they regulate the gene expression of housekeeping gene. Generally, it has been observed that housekeeping genes have low CpG promoters. So obviously they will be having low DNA methylation as well. So having high CpG content promoters, they generally observe more DNA methylation, and thereby they regulate the activity of housekeeping gene, that is option A. Amphibian eggs undergoes equal radial holoblastic cleavage, unequal radial holoblastic cleavage, equal radial meroblastic cleavage, or unequal radial meroblastic cleavage. As we know that the amount of yolk and its distribution in an egg decide the kind of a pattern cleavage is going to follow. If suppose an atypical amphibian egg which is having less yolk content, if we follow the cleavage pattern, then we will see that cleavage actually gets completed. It divides the egg into a complete portions, and that's where it has to be holoblastic cleavage. Now if this cleavage produce asymmetric blastomers of unequal sizes as we see it in the amphibians, then this would be of unequal holoblastic type. Now one more pattern we observe, that in amphibian eggs this unequal blastomers, they are gets arranged in a radial fashion, that means one over another. Hence cleavage in amphibian egg would be radial, unequal and holoblastic type, that means option B. Which substance determine the type of cleavage during development? Nucleus, cytoplasm, yolk, or A and B, that means nucleus and cytoplasm. As we have already discussed in the previous question, that the amount of yolk and its distribution pattern determines the type of cleavage. So option C has to be the correct answer. Which of the following are not true for Carl Ernest von Bayer's principles of law of embryology? A. General features of large group of animals appears earlier in development as compared to the specialized features of a smaller group. B. General features of a large group of animals appears later in development as compared to the specialized features of a smaller group. C. Early embryos of a higher animal is look like a lower animal. D. Early embryo of a higher animal is look like embryo of a lower animal. Now, out of the given pair of statements, we have to find which of the following is not true as far as Bayer's law of embryology is concerned. In 1828, Carl Ernest von Baer actually proposed 4 laws of animal development. So let's first understand what he has said. In his own words, the first law was the general character of the big group to which an embryo belongs appears earlier than the special character. The next law was the less general structural relations are formed after the more general and so on until the most specific appears. It is very difficult to comprehend these two statements. Let's simplify these. If you can recall this slide, as you know that whether it's a human, chicken, turtle, salamander or fish, the earlier embryo, they look like same. You cannot differentiate which of the following belongs to chicken while which of the following belongs to turtle. That means what? That the general feature of a large group of animal, they appear earlier in development. In early stage of the development, you cannot differentiate. All of them belong same. All of them has a almost same character as compared to the specialized feature. Specialized feature, if you can see, that means the features where you can differentiate whether it's a turtle embryo or human embryo, that you can observe it at the later stage of development. So definitely statement A is correct, that the general feature of a large group of animal appear earlier in development as compared to the specialized features of the smaller group. What about B? General features of a large group of animal appear later in development. That is incorrect. It appears early in the development. Now, what was his third statement? The embryo of any given form, instead of passing through the state of other definite forms, separate itself from them. And what was the fourth statement? Fundamentally, the embryo of a higher animal form never resemble the adult of another animal form. Now, again, let's simplify these two last statement about Bayer's laws of embryology. If you can see, if you have to predict which embryo belongs to human, which embryo belongs to turtle or a chicken or a salamander or a fish, you can do it at the later stage of the development rather than early stage of a development. That's very correct. Now, as far as later stage of the development is concerned, human never first become a fish or salamander or turtle rather than it becomes human. So that one thing is very clear, that human follows its own course of development. It never first become a fish or chicken or any other evolutionary lower forms. It always forms its own course of development, and that's where the early embryo of a higher animal never look like a lower animal. It only look like that its own version of adult at the later stage of development. So as far as statement C is concerned, that early embryo of a higher animal is look like a lower animal, no, it never look like an animal, lower animal. It definitely is a miniaturized version of its own adult. Option D, early embryo of a higher animal is look like an embryo of a lower animal. Yes, at the early stage of the development, all the embryo, they look like same. So whether it's a human embryo or a fish embryo, they look like a same. So early embryo of a higher animal, it is as same as like a early embryo of a lower animal. So which two statements are incorrect? Statement B and statement C. So here the option C has to be the correct answer. Enzymes involved in RNA interference are dicer, coenzyme, bicoid, or masking. As we know, the dicer is an enzyme which plays a role in the initiation of RNA silencing by recognizing double-stranded RNA and cleaving into small RNAs using its RNase 3-like activities, and thereby these dicer regulate the biogenesis of mi or si RNAs. So option A has to be the correct answer. CRISPR is. So there are four full forms are given and we have to find out which one represents CRISPR. So CRISPR actually stands for clustered, regularly interspaced, short palindromic repeats, and it is an important gene editing tool as a method of precise DNA editing for which Jennifer Doudna and Emmanuel Charpentier got Nobel Prize. So option A has to be the correct answer. Ramachandran plot is used for the determination of stable protein structure, also called as phi-psi plot, provide information about statistically favorable conformations, or all of these. Ramachandran plot, or also known as phi-psi plot, it's a two-dimensional graph which is used to predict statistically favorable or restricted conformations for a given amino acid sequence. And thereby, we may make a guess that what is the stable conformation which could be naturally present for a given sequence. So, Ramachandran plot is definitely used for the determination of a stable protein structure. It is also called as phi-psi plot, and it also provides information about statistically favorable conformation for a given amino acid sequence. So option D has to be the correct answer, all of these. Dextrans are polymeric glucose, polymeric galactose, polymeric fructose, or polymeric lactose. Dextrans are the glucose polymer having poly alpha D glucosides of microbial origin. So option A has to be the correct answer. According to Bohr effect, the oxygen transportability of hemoglobin is more when pH decreases, pH increases, pH remains stable, or it is not affected by the pH. According to Bohr effect, the low pH and high carbon dioxide concentration ensure that more oxygen unloading is being performed from hemoglobin. Actually, in the presence of high carbon dioxide, hemoglobin shows preferential carbon dioxide binding rather than oxygen, and hence oxygen actually gets delivered at the site. Now, as far as decreased pH is concerned, that means at low pH, the reversible binding of oxygen by replacing carbon dioxide is prevented. So both decreased pH and high carbon dioxide actually cause the oxygen unloading from hemoglobin. So option A is the correct answer. Cells are small in size because it provides more surface area to volume ratio. It provides less surface area to volume ratio, provides regulation of cytoplasm by the nucleus, or option A and B both. Actually, cells generally remain small in size to enable different regions of the cells to communicate with each other rapidly and maximize surface area to volume ratio for better diffusion of the substances. So in this case, cells are small in size because it provides more surface area to volume ratio. But always remember, there is another reason for being smaller in size as far as cells are concerned, so that they can well communicate with other cells. Protein purification in 2D gel electrophoresis is based on molecular weight, charge, number of subunits, or both A and B. In a 2D gel electrophoresis, in first dimension, based on the pi, or in simple words we can say, based on the charge at different pH, we separate the proteins. And now, by placing at 90-degree angle of these separated proteins, now we place them and separate them based on the molecular weight. So both molecular weight and charge is a criteria for protein purification in a 2D gel electrophoresis. DASH enzyme catalyzes conversion of fructose to fructose 6-phosphate, and it requires DASH as a cofactor. So is it phosphofructose kinase 1 or PFK1 magnesium ion, PFK2 or magnesium ion, PFK1 manganese ion, or PFK2 manganese ion. Actually here none of the answer is correct because the given reaction is catalyzed by fructokinase rather than PFK or phosphofructokinase. The reaction which catalyzed by phosphofructokinase is that the fructose 6-phosphate to fructose 1,6-biphosphate. So here none of the option is correct. Which of the following is not a possible catabolic fate of pyruvate? Lactic acid fermentation, citric acid cycle, ethanol fermentation, or glycolysis. Actually, pyruvate can enter into lactate or alcoholic fermentation during anaerobic condition, while into TCA cycle during aerobic condition. Actually, pyruvate is the end product of glycolysis rather than glycolysis is the fate of pyruvate. So option D is the correct answer. Allosterically regulated enzymes are oligomeric proteins, possess regulatory and catalytic domains, show sigmoidal behavior, or both option B and C. Actually, all allosteric enzymes are multi-subunit or oligomeric in nature. But all oligomeric protein need to be allosteric? That is not correct. They do have two types of sites, one where actually activity occurs and other which are the regulatory sites where modulator binds and regulate the main activity of enzyme. Remember, one of the major assumptions of Michaelis-Menten kinetics was that protein must not be allosteric. Hence, all allosteric enzyme shows sigmoidal curve rather than classical hyperbolic curve as observed for Michaelis-Menten kinetics. So, option D, that is statement B and statement C, is correct for the allosteric enzyme. Which of the following regulation occurs in slowest time frame? Feedback regulation, allosteric regulation, regulation through covalent modification, or enzyme synthesis through gene induction. Feedback regulation, covalent modification, and allosteric regulation. These are the three rapid methods of metabolic regulation, while enzyme synthesis through gene induction is a slowest mechanism which affects the biometabolic processes. So option D is the correct answer. The unit of maximum growth rate is hours inverse, hours, hours per liter, or hours per cell. Maximum growth rate or mu max is an important parameter which is present in Monod equation. Monod equation is an adaptation of Michaelis-Menten equation for modeling microbial growth under batch condition, where mu has a unit of time inverse, that means hours inverse. So option A is the correct answer. Mammals do not have ability to convert fatty acid to glucose because they cannot undergo glyoxylate cycle. There is no need for such conversion. They can obtain glucose from other pathways or both option A and C. Glyoxylate cycle has a potential to convert acetyl-CoA, which might be coming from fatty acids, into the glucose. This cycle is found in plants, bacteria, protistans, and fungi, and definitely absent in humans. Humans lack two important enzymes of glyoxylate cycle, that is isocitrate lyase and malate synthase, which can convert fatty acid to glucose. Hence, option A is the correct answer. Formation of glycogen cannot be initiated in the absence of enzyme glucokinase, glycogenin, glycogen phosphorylase, or glycogen kinase. Glycogen synthesis cannot be initiated without glycogenin protein, onto which the activated glucose molecules are tethered sequentially by the enzyme glycogen synthase. So here the option B is the correct answer. Glucose sensors utilizes dash as a bioreceptor. Glucose oxidase, glucose kinase, glucose dehydrogenase, or both A and C. Common enzymes which are used in making glucose sensor for glucose detection uses glucose oxidases, NAD dependent glucose dehydrogenases, FAD dependent glucose dehydrogenases, or glucose dehydrogenases PQQ. Out of all types of glucose related enzymes, FAD dependent glucose dehydrogenase is the most employed enzyme in glucose sensing industries. So option D has to be the correct answer, that both glucose oxidase and glucose dehydrogenases of various types can be used to construct a glucose sensor. Optical density is based on light refraction, light absorption, light diffraction, or light scattering. Optical density is a technique for evaluating the amount of light scattered by the particles suspended in a liquid sample. So option D is the correct answer, that optical density is based on the light scattering phenomena. Purity of DNA samples can be checked by measuring the ratio of absorbance at 260 and 280 nanometer. For a pure DNA sample, the ratio should be 1.8, less than 1.8, less than 1, or greater than 2. The 260 by 280 ratio of around 1.8 is generally accepted as a signature of pure form of a DNA. While if this ratio is around 2, then we can be assured that the sample contains purely RNA. If the value is quite less in favor of A260, that means that nucleic acid sample has a severe protein contamination. So here the option A is the correct answer, that a ratio of 1.8 of 260 by 280 is a characteristic that the DNA sample is pure. ATP molecules produced by the oxidation of one molecule of palmitoyl CoA to carbon dioxide and H2O are 38, 108, 148, or 40. Actually, palmitic acid is a C16 fatty acid, and after 7 rounds of beta oxidation, it will produce us the 108 ATP. As we know that before beta oxidation, there are 2 ATPs are used for the activation of free fatty acids. So hence the net gain would be 106 ATP, but the total ATP production would be 108. So option B is the correct answer. Carbamyl phosphate synthetase 1 or CPS1 is the first enzyme in urea cycle, and it is allosterically regulated by N-acetyl glutamate, N-acetyl glucosamine, N-acetyl galactosamine, or N-acetyl gluconate. CPS1 is a hepatic mitochondrial enzyme involved in the condensation of ammonia and carbon dioxide during urea cycle. This enzyme allosterically regulated by a chemical called N-acetyl glutamate. N-acetyl glutamate is synthesized by the condensation of acetyl-CoA and arginine, which is produced during the urea cycle. Now, higher level of arginine simply means that more N-acetyl glutamate will be produced, and hence there will be more check on the CPS activity. So, option A is the correct answer. Phenylketonuria is caused by dysfunction of phenylalanine hydroxylase, phenylalanine monooxygenase, phenylalanine mutase, or phenylalanine dioxygenase. Phenylketonuria, or also known as PKU, it is caused by the genetic deficiency of phenylalanine hydroxylase, which led to the impaired phenylalanine metabolism. So option A is the correct answer. Key protein involved in complement activation is C5, C8, C9, or C3. The central event in complement activation is the proteolysis of the complement protein C3 to generate biologically active products and the subsequent covalent attachment of a product of C3 called C3b to microbial cell surfaces or to antibodies bound to antigen. So the key protein involved in complement activation, whether it is coming from classical pathway or mannose binding ligand or alternative pathway, the central protein would be C3. So option D is the correct answer. Separation of components in gas chromatography is based on molecular weight, partition coefficient, molarity, or conductivity. The principle of gas chromatography is based on partition coefficient. The mixture of components to be separated is converted to vapors and mixed with the carrier gas or mobile phase. When these vapors interact with the stationary phase, they get partitioned between mobile and stationary phase as per their chemical nature. So option B is the correct answer. Pervaporation is ion removal, separation of liquid, separation of solid, or water purification. Pervaporation is a membrane separation process in which components from a liquid mixture are selectively transported through a microporous membrane. So option B is the correct answer, that pervaporation is a method of separation of liquid. Obligate anaerobes, that means strictly anaerobes, are not able to survive in the presence of oxygen because of absence of both catalase and superoxide dismutase, presence of both catalase and superoxide dismutase, presence of catalase but absence of superoxide dismutase, or presence of low levels of both catalase and superoxide dismutase. Obligate anaerobe, they are the microbes which do not require oxygen to live, and they would die in the presence of oxygen. They lack enzyme SOD, which is superoxide dismutase, and catalase, which are used for the conversion of lethal superoxide formed in the cell when oxygen is present. So obligate anaerobes are not able to survive in the presence of oxygen because they lack catalase and superoxide dismutase. Which of the following sentences is not true for pyrosequencing? It is based on detection of pulses of chemiluminescence, that is correct. Requires the activity of enzyme sulfurylase, that is also correct. Requires addition of dideoxynucleotide, that is incorrect. If you see it in the figure, we both require the pulses of chemiluminescence and also enzyme sulfurylase in order to perform pyrosequencing. But we never use dideoxynucleotides because dideoxynucleotide is a method or a step which is used in Sanger's method of sequencing rather than pyrosequencing. Incorporation of any dideoxynucleotide may further inhibit the action of DNA polymerase, as there is an absence of free 3' OH in dideoxynucleotide. So option C is the correct answer. Match the list 1 item with list 2 items. Translation, replication, ATP synthesis, and dark reaction of photosynthesis. Where do they occur? That's very simple. Translation, as we know, it occurs in the cytoplasm. Replication definitely occurs in nucleus because genome is present over there only, central genome. ATP synthesis, it occurs in mitochondria. That's where mitochondria is often regarded as powerhouse of the cell. And dark reaction, that means Calvin cycle of photosynthesis, it occurs in stroma of chloroplast. So if we collect, then option D is the correct answer, that translation which is occurring in the cytoplasm A24, replication which is occurring in nucleus B21, ATP synthesis which is occurring in the mitochondria C22, and D has to be connected with 3, that is option D. Impermeability of mitochondrial inner membrane to most ions, electrons and protons is due to the presence of cardiolipin, collagen, chitin or cellulose. As we know that the inner mitochondrial membrane is largely impermeable to charged ion, due to the special lipid molecule constituting it. Whereas the normal phospholipids are having only 2 acyl chains, while cardiolipins has 4 hydrophobic acyl chains. And that's where, due to the increased hydrophobicity, it is very difficult for a charged ion to make it through the plasma membrane of a mitochondria. Hence, it is the cardiolipin which ensure the impermeability of mitochondrial inner membrane to most of the charged species. Option A is the correct answer. Lateral root primordia are developed from pericycle, endodermis, ectodermis, or xylem. Lateral root primordia arises by the localized cell division in the pericycle, which resulted in the moon of the tissue. So, option A is the correct answer. Agrobacterium tumefaciens is used for the transformation in plant tissue culture because both agrobacterium and plants have similar genetic makeup. That is incorrect. It cannot be correct by any means because agrobacterium is a prokaryote and plants is a higher eukaryote. We cannot expect it to have the same genetic makeup. Option B, agrobacterium genomic DNA transfer its genetic material to the plants. No, it is also incorrect. Agrobacterium actually transfer a plasmid, that is Ti plasmid, not even full complete Ti plasmid. It's a part of the Ti plasmid which is being transferred to the plant genome. And upon integration, this tDNA, it produces a special chemical called opine for its nutrition. Actually, the transfer of this tDNA is facilitated by the expression of Vir operon gene. So, option C is the correct answer, that the Vir genes of agrobacterium aid in the tDNA transfer to the plant. Mutation in the class A gene in ABCD model of flower development leads to which pattern of floral organization? Arrangement of flower organ in a pattern in Arabidopsis plant is regulated by a set of homeotic genes like A, B, and C. When any of these genes is mutated, the original or wild flower pattern gets disturbed and changed. So let's understand what is this change is all about. If we see the wild flower, the arrangement of the whorl will be sepals, petals, stamens, and carpels going inward when both A, B, and C homeotic genes are expressed. If suppose A genes is mutated as asked in the question, then the original whorl will change to the carpel, stamen, and carpels. That is option C. Now, if B gene is mutated, then original whorl will change to sepals, carpels, and carpels. While if the C gene is mutated, then the whorl will change to sepals, petals, and sepals. So here the option C has to be the correct answer, that when the A homeotic gene is mutated, then the original whorl, which has sepal, petal, stamen, carpels, change to carpel, stamen, carpels. With this, we conclude this video. Please do subscribe us in large numbers. If you like our content, like, share, and comment on the video. Thank you very much.